Thank you so much. This is the result of a long-term collaboration I've had. Andy Gardner is my former postdoc, Kelly Shepard at the Western Australian Herbarium, Emily Sess at University of Florida, and my co-PI in this NSF-funded project, Diana Howard at St. John's University. So I'm the plant outliner in this session talking about phylogenetics and floral symmetry evolution of the core Goodeniaceae. All right, and I'm going to argue to you that Goodeniaceae is potentially an emergent model system for studying floral evolution in particular. If you ever have a Darwin quote, this is the place to use it. I am not the first person to have been tormented by this family. In fact, Charles Darwin himself studied the key synapomorphy of the family, which is this extracellular tissue that's involved in secondarily packing pollen in the bud and presenting it to pollinators, and its potential role in promoting outcrossing. And so he wrote in a letter to Hooker, "The Goodeniaceae have weighed like an incubus for years on my soul, so hopefully he's resting in peace a little easier. I've taken that burden." This family is a close relative of the Asteraceae, which is one of the largest and most important angiosperm families. So potentially things we learn from them can apply here and vice versa. This is predominantly an Australian plant family. It's in fact the 9th largest plant family within Australia, though there's been multiple dispersals out of Australia and subsequent speciation events, particularly in Hawaii. Within Australia, there's relatively high endemicity, particularly in the southwestern corner, which is a global hotspot of biodiversity. So kind of my first approach within this was doing, of course, a phylogeny of the entire group. And we readily identified two major clades, the largest of which we termed core Goodeniaceae, which is a single species sister to two larger clades. And in particular, this Goodeniaceae group here has the most species, the largest genus Goodenia as it stands now, and a lot of taxonomic complexity within it. I've been very fortunate to be funded by a National Science Foundation, and our working group labs focusing on Evo, Devo, and systematics are, one, to produce the best sampled and best resolved phylogeny we possibly can of that Goodeniaceae clade, supporting an actual taxonomic revision of this group. It's clearly needed. And then to study the evolution of floral symmetry across that broader core Goodeniaceae group using comparative and Evo-Devo methods. Okay, so phylogeny first. We start with tons of Sanger, and I'm particularly proud of our coverage and inclusion of taxa. We have upwards of 95% of the 200 plus species in here, as well as a whole bunch of potential new species. It's kind of cattle all across Australia. If you got something, throw it in. We're doing the big phylogeny. We have things placed very well into big clades. A lot of smaller genera are independently very well supported with molecular data, as they have been historically, with morphological as well. The more taxa we've added, of course, it gets more and more complex taxonomically, especially at the subgeneric level. So to kind of simplify that big picture, this is what Goodeniaceae looks like. So there's several well-supported monophyletic smaller genera, that big genus Goodenia is in 3 big clades, embedding some other things as well. There's strong support for each of these lineages independently, but as we saw in the last talk, we again have a very poorly supported backbone, emblematic of, again, an ancient, probably rapid radiation. So instead of spending a ton more time and money adding more and more loci that probably wouldn't do it, we opted for a hybrid kind of next-gen approach. So we selected 24 taxa targeted across all of these clades for genome skimming, next-generation sequencing technology, and first started with the chloroplast, kind of the workhorse of plant systematics, and we've actually moved into the mitochondria with a little bit of data from different nuclear loci as well. Okay, so we assembled the CDS of the chloroplast for those 24 and concatenated it to that broader Sanger data set, and it solved most of our backbone problems. We had really good support for most of those deepest nodes, supporting monophyly still of these major clades and giving some order. With the perennial problem of this Goodenia clade C that I'm going to talk a lot more about. Meanwhile, looks like this. She's probably asleep now, but in 12 hours or so, she'll wake up and go about the task, the hard task, of trying to find morphological synapomorphies for most of these new clades that we are going to be circumscribing as genera. Now, in many cases, we are just reinstating older names. For example, Goodeniaceae clade beetle Phylogenetics. And so the most complex thing that she spends most of her time on here is what are we going to do with this clade C? We are really trying to retain existing genera as well as authoring new ones as well. Okay, so here's a closer look at clade C. 5 big clades, enormous morphological and taxonomic diversity here. This is fascinating when I'm wearing my evolutionary biology hat and terrifying for her as a taxonomist. And again, very poor support on the backbone there, even with the chloroplasts. So we were like, well, what can we do next? We invested a lot of money in our next-gen sequencing, and so we dived back into the pool, and we've actually moved to looking at the mitochondria. We actually did a second round of next-gen as well, so that we had even better sampling within here. And I'm not talking about it today, but we've done a whole bunch of SH tests as well. Animal people out here, you might be a little surprised to know that mitochondria in plants are typically not used sequences for systematics. The adage has been that the sequence evolution is relatively low, whereas the structural evolution is very, very high. But this might vary a lot, actually, by taxonomy. It may actually be very appropriate in some cases for giving resolution. I'm going to tell you about my student Price's. He has a poster all about this tomorrow night, if you want to learn more. So what we found in this case, we assembled about 50,000 base pairs of the mitochondrial genome within those 24 as well. And we found in general a very highly congruent story between chloroplasts and mitochondria, except within clade C, where we needed it, of course, right? And we did find, though, that mitochondria in this case was as variable, if not more variable, than chloroplast was, which was exciting. So that right there, basic clade C, is the incubus on my soul, still moving forward, more and more study there. And there is a hard incongruence in the positioning of some of these that is statistically significant. So something's going on there that demands more study. Polyploidy reticulation, something might have happened there. Okay, floral symmetry. These are really stunningly beautiful flowers, but I'm obviously aware of who they're talking to, the pollinators of Australia, right? So some observations about floral diversity in this group. 2 sister clades, Scaevola, Goodenia. Scaevola, fewer species, shorter branches than its sister Goodenia. And for the most part, all of the flowers are 5 petals to one side, fan flower. It's fairly rare in angiosperms. In contrast, in Goodenia, there's a lot of lability. You get kind of bilaterally symmetrical in many different ways, as well as the repeat evolution of the fan flower multiple times within Goodenia itself. We did a historical biogeographic study of this as well. I won't belabor it much, but here is when that Scaevola, the fan flower lineage, its kind of crown radiation happened in the mid-Miocene, coincident with the repeat evolution of the fan flower within Goodenia as well. Okay, so we really want to get to the point of studying in a comparative method all this floral symmetry across the broader group. But the first thing we needed to figure out is how can we objectively categorize floral symmetry? And then can we test mathematically, is it true that Goodenia sensuatus shows a higher lability in floral symmetry than Scaevola? And Dianelle, my co-PI, is really interested in the cycloidea gene family and how history, potential of duplications and expressions is responsible for these different floral forms. So to get at creating categories of floral form, we undertook a 2-dimensional geometric morphometrics approach. So what we did is, for 44 species within core Goodeniaceae, we took lots and lots and lots of head-on shots of the pictures in the field all over Australia, and then applied landmarked points to the homologous tips of the 5 petals. And then subjected those images to a Procrustes transformation, which corrects for angle, size, things that might have varied. And then made a consensus per species, and then compared those different species to each other. And then we did a PCA analysis of all of that. And we actually found that only 2 PCAs accounted for 98.5% of the total variance of these landmark points, which is pretty exciting. And PC1 in particular, which is responsible for the movement of these dorsalmost petals, was the lion's share, 86.6. PC2 is the lateral petals, how they move up and down. Okay, so then to test that hypothesis, is Goodeniaceae sensu-latu more labile than Scylla sensu-latu? We compared those PCs and said yes. In fact, Goodeniaceae sensu-latu does have higher variance in both PC1 and 2, as we predicted, and an overall greater average dorsalization. The repeat evolution of the fan flower can probably account for a lot of that. We then wanted to see if we could make clusters or categories that then we can apply in a comparative framework across our phylogeny. And we see a nice kind of clustering here of, again, those fan flowers. So you could see with your eyes and subjectively say this is a fan flower, you would probably be correct placing it in here. This is the axis of PC2 here, and it's a little harder to find a nice cutoff between bilabial and pseudo-radial. We have kind of these rules with PCs and how they scale that you could put things into different categories that way. So what comes next is actually a really cool project where we've crowdsourced head-on images of as many species as we possibly could get, and we're landmarking them and applying this, and we'll study both discrete and continuous aspects of this in a comparative framework. Okay, then my colleague Dianella Howarth's really interested in the Cycloidea gene family, which we know is really important in bilaterally symmetrical form of angiosperm flowers. So she's been working a lot on this Jabaily group. There's some gene silence she's done, and you see you get this kind of ancestral radial look. So there's a lot that's known about this gene family in angiosperms. So we've been taking data from both plants that you can get here, like horticulturally, as well as down in Australia, to look at the levels of expression of homologs within each petal position in species differing in symmetry. Okay, so first looking at gene copies distribution across the entire family. There's 3 well-supported copies of Psych, and this also includes that sister clade in Goodeniaceae, the lad clade. So we don't see any specific duplications within core Goodeniaceae or within Goodeniaceae sensu-latu. We do see a duplication independently in Goodeniaceae within this copy, Psych3. And this is actually kind of the cool, potentially, story that's going on here. Okay, so here's some qPCR results from Scaevola emula. This is a fan flower you see planted horticulturally all over in the US. It's really the only one that's available easily, which we've learned is a very hard thing to do evo-devo work on things you cannot grow and don't live here. But at any rate, Psych2 behaves as we see in other angiosperms, very strong dorsal expression. In stark contrast, Psych3A and 3B is strongest expression in the ventral petals, which has apparently not been seen in other angiosperms up till now. So that's potentially really exciting, and we're very excited about results from other species in the core Goodeniaceae, those independent derivations of the fan flower. What are they going to show? Bilateral symmetrical petals as well. So with that, I really want to thank the National Science Foundation for supporting this work, showing what you can do at a liberal arts school with really amazing undergrads. Undergrads have been central to all of this in my lab. Tons of collaborators, all of Australia is on board supporting us, particularly with the taxonomic work. Great undergrads, grad students, postdocs at St. John's, and University of Florida for their expertise and their computational power. Thank you very much.